Welcome to our poster, Kinetics and Potency of GD2 CAR-T Cell-mediated cytolysis of glioblastoma. Glioblastoma is an aggressive form of brain cancer with no effective treatments and a short prognosis. Immune cell therapies offer promise, and targeting tumor-specific antigens with chimeric antigen receptors can lead to high specificity. Axion BioSystems Maestro Z platform is an impedance-based system for real-time, label-free, continuous monitoring of cell viability and cytolysis. Impedance is measured from electrodes embedded in the bottom of each well of a 96 or 384 well plate, and as cells attach, the impedance increases, and then if cells die or detach in response to dosing, impedance decreases. Here we use the Maestro Z to assess the potency and kinetics of immune cell-mediated killing of glioma stem cells, a subpopulation of glioblastoma, by GD2 targeted CAR-T cells over the course of 7 days. First, patient-derived N08 glioma stem cells were assessed for CAR-T targets using flow cytometry. Glioma stem cells exhibited high expression of the antigen GD2 compared to other popular CAR-T targets. High GD2 expression was also seen in rodent xenograft tumors and human patient tissue arrays compared to healthy tissues. Next, patient-derived N08 glioma stem cells were plated at 50K cells per well on cytoview Z 96 well plates, and their impedance was continuously monitored on the Maestro Z. As the glioma stem cells attached, their impedance increased, while media-only wells shown here in red showed little change. At 48 hours, GD2 targeted CAR-T cells were added at effector to target ratios ranging from 0.1 up to 10 to 1. Impedance and cytolysis were then subsequently monitored for up to 7 days. In addition, some wells were left untreated. These are the glioma stem cells alone, shown here in teal, to serve as a no treatment control, while others received 50K CAR-T cells alone, shown here in pink, and because these don't attach, they show little change in impedance. All ratios of GD2 CAR-T cells, shown here in green, purple, and blue, induced a decrease in impedance that indicates glioma stem cell death. Higher ratios of GD2 CAR-T cells caused an initial increase in impedance that's likely related to cell swelling or inflammation before then inducing rapid GSC cell death. Because impedance is nondestructive and label-free, impedance can also be used to measure not only endpoint potency, but the kinetics of cell-mediated killing as well. Up here we present percent cytolysis. 0% cytolysis is equivalent to the impedance of glioma stem cells alone, so untreated cells, whereas 100% cytolysis is equivalent to full cell death. We compared the cytolysis induced by naive activated T cells compared to targeted GD2 CAR-T cells. And as you can see, targeted GD2 CAR-T cells showed greater potency for killing glioma stem cells, with all ratios reaching greater than 50% cytolysis. Interestingly, while the final percent cytolysis did not differ across ratios of GD2 CAR-T cells, the kinetics did, and that's shown here over time. The blue, the 10 to 1, the highest ratio, actually showed a delayed cytolysis, again likely due to that initial swelling, and a slightly longer time to reach 50% cytolysis. Finally, GD2 CAR-T cells were evaluated for activation and early exhaustion after 2 and 7 days in co-culture. After 7 days in co-culture, GD2 CAR-T cells exhibited markers of chronic activation, including high CD69 and increasing CD137. Early exhaustion of GD2 CAR-T cells was also suggested by expression of PD-1 and LAG-3. Overall, the Maestro Z platform enabled continuous, dynamic, label-free quantification of the potency and efficiency and kinetics of immune cell-mediated cytolysis of glioblastoma. Our results suggest that GD2 CAR-T cells are a promising treatment for glioblastoma.